So I am talking about current trends in regulatory safety testing with the in vivo comet assay, and my name is Marie Vasquez. I'm with Helix 3, a small CRO that specializes in comet. Just to begin, to let everyone know, I know most people think or consider in vivo safety testing is mostly pharmaceuticals, but it actually influences a lot of industries, including industrial chemicals, consumer products, agrochemicals, food additives, even medical devices and supplements. And involves several regulatory authorities, both within the US and in the EU. These are just a few of them that I come across or work with directly for safety testing. And just so to give a little bit background of the difference of OECD TG 489, the testing guideline for COMET, and the applicability across the industries can be a little complex, as you can see with pharmaceuticals. Starting out, you know the intended human exposure, you know the exposure route, the pharmacokinetics of your compound, the effective or intended dose, and even have some clinical data. But as you move into the other industries, such as consumer products, you don't necessarily know about whether there is intended exposure or known exposure route. For example, until very recently, it was never considered a concern that anyone would be ingesting bleach, but now we have to take this into account. For the same consumer products, such as cleaning products, you would definitely not have pharmacokinetic data, again, because it was not intended for human consumption. You may not know the effective or intended dose, and you may not have any clinical data at the time you go into in vivo testing. Industrial chemicals, they're definitely not intended for human exposure. There's multiple possible exposure routes from inhalation to ingestion or oral to skin. There's rarely, if ever, any pharmacokinetic data, again, since it was not intended for human exposure or ingestion. And there's no effective dose and no clinical data. And many times you're having to deal with environmental exposures. Medical devices, that's even more creative because you know the intended exposure of where this device will be implanted, but you don't know the exposure route necessarily or what degradation products might be released, how or where or when. And you may have some clinical data, but you won't have necessarily any effective or intended dose because it's not a liquid compound. So from a regulatory perspective, stating the obvious, that their primary focus is protecting human health and optimizing or minimizing the use of animals. But for the purpose of this talk, I'm going to just talk about their concerns or focus, which is assessing risk for evolving industries and exposures, evaluating risk in specific tissues and organs, and understanding the mode of action for many compounds in safety testing. From an industry perspective, again, their focus obviously is ensuring product safety and profitability, but that is a factor to consider, as well as optimizing and minimizing the use of animals and resources. And then, of course, from their perspective and for the purposes of this talk, their interest is addressing regulatory risk assessment requirements, minimizing risk in specific tissues and organs, and understanding the mode of action, very similar to the regulators. So the first topic, evolving industries and exposures, the testing goals for that is specifically to look at compounds unintended for human consumption, such as consumer products, as we discussed earlier, new products that are getting developed every day, some of them, for example, including conjugates, and different routes of exposure, such as inhalation, even ocular exposure for some compounds, and unanticipated exposures, as I mentioned, environmental exposures where they're in the drinking water, the groundwater, or even when they're on dermal exposure. The issues with testing the evolving industries and exposures are, as I mentioned earlier, again, there's limited or no in vivo or ADME data, absorption, distribution, metabolism, and excretion data. You're also testing compounds that are highly cytotoxic and corrosive, and for that same reason, they may not be very bioavailable. And this presents a lot of study design and logistical limitations with how you can do these exposures for safety testing. So the goal for evaluating specific tissues and organs is you want to look at compound-specific target organs, whereas previously or pre-comet, you were limited in vivo testing to bone marrow and peripheral blood, but now you open this up to where you can look at any species, brain, sperm, pancreas, any organ. So you can also open up the safety testing options for identifying possible germ cell mutagens. And you optimize a list of the use of animals and resources because you can test all of these and evaluate multiple tissues in the same animals. But when you're evaluating specific tissues or multiple tissues and organs, there are testing issues. For example, how and which organs, how many organs do you test? It can get a little out of hand when you say you want to minimize the use of animals, but then you're also evaluating a lot of tissues. You also have to take into account the influence of cytotoxicity on some of the results, where it may be cytotoxic at the site of contact, for example, in the GI tract. You still also have to test in the liver. So you're limited by the cytotoxicity at the site of contact for the dose you can select. The other limitation is establishing proficiency. You have to have proficiency with each tissue. They are very different. Starting out, most people are used to testing in the liver and maybe the GI tract, but as you get into some of the other tissues, there is a little bit of special technique in optimizing for each tissue. And with that comes establishing historical control data for that. And at the same time, again, you need to minimize the use of animals and resources. It's difficult to establish historical control data in every tissue possible that could be tested. So several of the examples I'm going to give you throughout this talk are mostly from EFSA and ECHA. I just want to point out I'm not picking on them. They're just the nice ones that actually publish most of their decision letters online, so they're publicly available versus FDA, which they're not. So that's why their examples are mostly provided here. But on the topic of which and how many organs to test, EFSA, this is an example of one of EFSA's decisions or recommendations and the questions regarding the target organ. If the compound did not reach the target organ, then they are looking at considering the site of contact. And of course, the test guideline specifically says you should consider, ADME data, absorption, distribution, metabolism, and again, excretion. And these are some examples. As I said, most people are used to considering the site of metabolism as the liver and the site of contact as the GI tract, but you have different tissues that are now optional. Some compounds are actually metabolized in the spleen. Some will be absorbed, or site of contact may be the cornea. The distribution, it could go to the brain across the blood-brain barrier, and excretion, you can be looking at the bladder. And as the mechanism of action, some compounds that directly interact with an organ, for example, such as the pancreas, which is there's limited data on the pancreas. This is an example where EFSA has actually stated that because there was not systemic exposure, or there was insufficient plasma analysis exposure to show that the bone marrow did not necessarily get exposed. So therefore, they did recommend the in vivo comet. And this is just one example of where the in vivo micronucleus assay, although that was been highly recommended as a follow-up to in vitro, it may be insufficient if you don't have adequate systemic exposure. And in this case with ECHA, they are suggesting the site of contact as the gastrointestinal tract. And in many cases, in most cases actually recently, they're asking both for the glandular stomach and the duodenum, not just one, because they may potentially act differently with exposure. And in this particular case, again, for an inhalation compound, they are recommending multiple tissues within the respiratory tract, the nasal mucosa, the alveolar epithelial cells, and bronchoalveolar lavage cells. And in many studies now, they are starting to also request for possible germ cell mutagens that if any compound is positive in a somatic tissue, that they also want to evaluate gonadal tissue. Because there is complexities with evaluating sperm or the actual germ cells, because at what stage of development, that is still in the works of what stage of development. So right now, the best test they have available to request is gonadal tissue, because that at least gives you some indication that the germ cells were exposed if the gonadal tissue is also shows DNA damage. On the topic of influencing cytotoxicity on results, I give you just an example from a book chapter. This is a picture of the duodenum where this is healthy tissue, and this side here is where it was test article exposed, where the epithelial layer is almost completely removed by the test article exposure, and where in this case you have almost no epithelial tissue, you have villi blunting, and in this particular case, DNA migration was actually decreased compared to the vehicle control because this tissue, the cells that you would normally have raising the baseline level, the epithelial cells are missing. Here on the left are some images of the infamous hedgehog cells. These were actually created using the acellular comet assay where nuclear DNA was exposed directly to test compounds. So this is not due to cytotoxicity. This is pure genotoxic effects you're seeing. And you can see migration patterns as high as this cloud. And this is something that you're having to take into account in vivo. And on the right side are examples of, this is liver cells, liver tissue, where you see glycogen depletion is actually correlates with low molecular weight DNA diffusion, where you take comet slides immediately after 1 hour in lysis with no electrophoresis. And with different stages of diffusion, you can see low molecular weight DNA migrate or diffuse away from the nucleus and developing this pattern. So this, where they're almost faded out, correlates with very low glycogen in the liver. And they also, when you add electrophoretic conditions to these, they disappear. Or for example, this may look like this when you do electrophoresis. So this is just an example of where industry has published or has been published the concerns about evaluating GI tract, that it may give a false positive. That's the concern. Right now, you see an increase in DNA migration at this high dose, where this laboratory specifically did tunnel staining for apoptosis and concluded that the stomach was most likely, the increase in the stomach was most likely due to apoptosis and cytotoxicity in that tissue, and not necessarily a genotoxic effect. However, you have to be very careful about whether this argument will be accepted by regulators. In some cases, you're going to have to defend it very well. The previous lab seemed to do that. In this particular case, there was only cytotoxicity evidence of it by histopath in the non-glandular stomach, but the DNA migration increase was noted in the glandular portion, and so it was not considered relevant to the results in the glandular tissue, even though it was the same organ technically. On the topic of establishing proficiency, this is just another example from EFSO where they cited that the laboratory said the test article dose values were within the historical control negative range, but their historical control data range went from 0.05 to 7.14% for percent tail, which is quite wide, and therefore they discounted the use of their historical control data range to qualify this result as negative, and instead said it was positive. And as I mentioned, when you have, for establishing proficiency in historical control data, in order to get the variability down and across multiple tissues, when you expand how many tissues or consider how many tissues COMET can actually evaluate, this becomes very labor intensive and a costly project to validate in every one of the tissues that are optional and every one of the species and strains that are options. And so this becomes a matter of minimizing the use of animals and resources. How do you do that when you have to establish in all these tissues that are possible for your compounds? So another testing goal is to understand the mode of action. And this is an example where ECHA has specifically requested the use of the enzyme modified COMET assay to look at oxidative damage in vivo studies. And they are currently requesting this in some cases. There's also been cases where they've asked for evaluating the cross-link induction. Now, both of these are not mentioned in TG489, but they are still able to request this option looking at certain compounds with certain profiles. But the issue with understanding the mode of action, the testing issues, mostly revolve around logistics. So logistics of applying these assays to animal studies the problems include limited possible dose groups, because you can only have maybe 3 test article dose groups so that you don't have an option to do wide ranges to minimize use of animals. You have multiple cell types, more tissues evaluated all at the same time, and you have time constraints of multiple sample collection and processing where sample collections could span multiple days for one study. So you're and all day long. And then you also have the requirement for specific to comet that you have to have a balanced sample and slide processing requirements to limit variability in artifacts that can appear to look like a dose response if you, for example, process all of the positive controls on day 1, all the negative controls on day 2, all the high dose on day 3, and etc. Just to give you an example, so when you start out with a study, like I said, a standard study would say 3 test article dose groups, 1 positive and 1 negative control group, and 6 animals a group. You start out with 30 animals. Just taking a standard 3 tissues with liver, stomach, and duodenum, you already have 90 samples. You have to make 3 replicate slides a piece, so you end up with about 270 slides. Then when you have to electrophoresis them, this is based on our gel boxes at Helix, where we can fit 30 slides, up to 30 slides per gel box. So that still breaks down to about 9 electrophoresis runs with 2 hours each run. And what you have to consider is that in every gel box and run, you have to have every dose group represented, all the tissues, all the samples for one tissue run together on the same day, and a random distribution of slides to make sure that you don't have any artifacts appearing to be a dose response just based on the position within the gel box. And so again, you also have to have all the samples of the same tissue must be run the same day, including additional comparative slides that may be enzyme treated, just because you want to make sure that one set doesn't sit in lysing longer than another set, again, creating a potential artifact or what that could appear to be a dose response of some sort. So you can see you add one more tissue or you add one more process and this grows, this takes all day long. And now you start running into the problem of logistically, how do you do this? If you have a study, like I mentioned you incorporate it into a toxicology study where necropsies span a week with one set of necropsy every day, how do you process all of these to make sure that every slide and every tissue is in lysing at the same amount of time and electrophoresed at the same time so that there's minimal time variance? So for another issue with understanding the mode of action is the regulatory requirements. They also require a positive control for each type of damage in every tissue evaluated. So if you, for example, are looking for oxidative damage, then you have to make sure you have a positive control that you know will induce oxidative damage in vivo in every tissue that you're evaluating. You don't get the option of selecting just one tissue, it has to be in every tissue evaluated. That can be quite complicated, which I'll get to later. You also have to have strict quality control of reagents, including the enzymes. So this could be another issue with controlling that quality. And then in many cases, you also have to have validated methods so that if you get a negative, that you can prove that your methods were adequate for actually detecting an effect in the first place. So in order to do that, you must have adequate control of variability, which again takes into account the designing and logistical issues we just mentioned. So on the topic of positive control, this is an example that Gunter Spite published a few years ago in 2012, and this is regarding formaldehyde. And one of the problems with cross-linkers, at least in this particular case for formaldehyde and in our experience with cisplatin, some of the cross-linkers tend to induce cross-links only at the site of contact, and then they do not go systemic at that point. So you may have to have, unlike EMS, where you can use it as a positive control that induces damage in basically every tissue with one same dose in the same animals, you may have to have a separate positive control for each tissue for cross-linking, whereas you use just the lungs or the basal tissues within the lungs for the cross-linker, but then you may have to have a separate cross-linker that induces cross-linking in the liver, for example. That means you would have additional dose groups and then additional animals required for each type of damage and tissue evaluated. When we talk about the strict quality control of reagents, and I just honed in on enzymes just because that's specific for the reactive oxygen damage assay, there are several publications that have come out that have actually stressed that, at least as far as I know, and recently, the enzymes from commercial suppliers still have not yet been validated. And so these publications are recommending that you do titration experiments for each batch and each supplier before you use them, because they can vary between manufacturer's batch and even cell type. So you do have to do titration experiments for each before starting. And that can be, again, quite complex trying to incorporate for in vivo studies and safety testing, when you have to do that in advance and taking into account all the tissues that you're going to evaluate. So although there were issues, there are extreme benefits to having these methods incorporated into in vivo testing. One, they will allow for the sensitive detection of mutagenicity enhancing damage in vulnerable organs, something that we haven't had access to pre-comet. Again, looking at the pancreas, for example, where pancreatic cancer is a big concern because you don't get much symptomatic warning in advance until it's too late. We may be able to detect damage in the pancreas that could be an early detection indicator for cancer risk. You would also have more informed risk assessment for existing and new compounds and exposures as we develop these, because again, COMET is flexible, so it can take into account some of these new exposures that were not previously considered before, for example, the industrial chemicals that are now being retested through REACH. It would also allow a better understanding of mode of action for regulators and for industry. Some compounds, if you understand more about oxidative damage, for example, it could provide possible risk thresholds, which would allow you to make more informed decisions about exposure risk. So how to help? I encourage all of you to attend discussions in the COMET project for HESI, the Genetic Toxicology Technical Committee in Vivo Work Group. We are currently working on a COMET project that are looking at these specific issues and working closely with regulators to come up with recommendations for how to optimize the COMET assay for safety testing. Many of you have a lot of experience with these assays in vitro, and we could really use your help in this project. And if you are in academia or any government program, you can join HESI for free, unlike commercial clients or commercial companies. So I encourage you all to attend and join them on this website. And also, if you can help us develop methods to help address these logistical and quality challenges, because I believe they can be fixed. We just have to work together to come up with a way we can do it. And then lastly, I just want to thank the regulators who have been contributing to this. They are very involved with our project and have been really providing useful information and input from their standpoint and perspective to help us develop the safety testing methods that would be beneficial to everyone. Thanks, everyone.
Thank you, Marie, for this very interesting talk and a little bit different perspective. Do we have questions? Please, if you do, type them in a Q&A box. You can do it.
Yes, Roger?
Yeah,
Roger.
I cannot type in the Q&A box, unfortunately. I have a question about the use of enzymes. You said it's very important to get more information about the mode of action. So by using specific enzymes, you could say something about the type of DNA damage that is there. And does that help in the mode of action?
Yes, yes, it does. This has become quite a bit of interest because one is concerned whether your compound causes direct oxidative damage or is it secondary to an inflammatory response. It's early. So that's one thing that they want to understand, because in many cases, if it's just an inflammatory response, it's not necessarily genotoxic, or again, it could be a threshold where your body can repair that damage. And is it something that we need to understand if your body, one, becomes overwhelmed, at what stage, is the system overwhelmed? So yes, it is very important.
Marie, maybe your opinion, similar question as Boana. Is it possible for 3D cell cultures to in the future replace maybe animal models in experiments, or?
I don't think it will replace. I agree with her that I think that it would enhance so that we could minimize the use of animals and focus them more appropriately, but I don't think it will replace it. I think it's still very difficult because in the animal, so many things within the body, the organs interact, the cell-to-cell interaction, circulation, things that cannot quite be replicated. I think in culture, I think that's a valuable information that we need for before you go into humans.